So a couple of important things. Next week is the test. Right? That's big time important, right? So what I was doing last night was putting some finishing touches on some stuff for you guys. So to bring to your attention first is, remember those practice quizzes I had posted for you guys in the past, right? They're practice. You can do them as many times as you want. So I highly suggest, obviously, going back over quiz 1 with terminology. Quiz 0 with terminology from Lab 1. Identifying eukaryotes and typing out their names, because you do have to be able to spell them, right? And identifying them, as well as answering some questions referring to the eukaryotes. This is the part of the test 1 that everybody hates, right? Because it's a lot of memorization, right? So, and then I just added a new one called Staining ID Quiz. And again, these things have points associated with them, but not really, right? Because you don't really get these points. But just to give you an idea of like how you did, kind of thing. Does that make sense? Right? So you don't get these points. And even though these had deadlines, you can still do them again. Okay? So they're still open to you. They're not closed. So the other thing I do have for you that does count for points, as you'll notice that under the Exam 1 folder, and obviously I want you to do this before the exam, which is March 3rd. I want you to do by March 1st, take this quiz. And then after 11 o'clock on March 1st, when you click on it, it'll tell you the answers, right? So you'll know what you got right and wrong and why, and this will help you study for the test. And this is staining result ID. So I'll show you a picture of a stain, and you have to tell me what the result is. Is it gram-positive? Is it gram-negative? Is it acid-fast? Is it non-acid-fast? Does it have a capsule? Does it not? Does it have a spore? Does it not? Right? What's the shape and arrangement? And it's multiple guess, right? In other words, multiple choice. And technically you're at home, right? So you can have your notes open, right? This should be an easy 10 points. Okay? Counts as quiz 6. You got to do it by March 1st, so that way, that day too, you can see your results and you have a day and a half to come see me if you have questions. Okay? All right. So the other thing is, the day of the exam, we are going to prep for the next lab. You guys are going to set up your plates for Kirby Bauer. So I found a pretty good video, because I find even when I demonstrate for you guys, right, it's hard for you to see. So I found last night like 1-minute video, kind of like how I did for you guys with focusing the microscope. What I will do is we're going to set up, we're going to inoculate those plates, and then we're going to clean up. Then I'm going to have you guys step out for a few minutes, and I'm just going to put my questions on the tables, and then you'll come back in and take the exam. Okay? So for that demonstration, though, what I'm going to do, and if you want to look at it ahead of time, which I'm sure you probably won't because you're going to be concentrating on the test, and that's quite all right, I'm just going to put it up on the screen so everyone can see, right, how to inoculate these plates so we don't goof them up. And we'll all, inoculate plates, including me, because we need 4 sets. Each table is going to do a set, right? So we kind of lost a table, so I'll do that set, right? And then I'll incubate them, and when you guys come back next week, you'll be able to go right into measuring and getting your results from running that test. And that'll make sense after the test when you actually do the homework assignment related to what you set up. But you can set it up, right, without knowing all the particulars of what you're doing, right? You just need to follow a protocol. Okay, so that's why that is there. And that really is there. Right, really is there. And so I have fixed, unfortunately, it wasn't until this morning, right, the notes and these videos may help you for reviewing for the test. So I did put those up, and I apologize that they weren't up ahead of time. So we're going to go into first, and we've got a shortage of paper going on right now Otherwise, I would have printed this for you, and hopefully some of you guys may have printed it. But you don't necessarily have to print it, right? It's on Blackboard. Wow, I said the B word. It's on Canvas. You have access to it whenever. So the first thing that I'm going to do is go over this document called Staining ID. And I have it, of course, in a PowerPoint with all the answers, which I'll post that for you here as well. And then I'm recording, right? So you'll have access to all this. Usually sometimes I post it ahead of time like I did this time, because obviously it's in color. Some people like to print stuff in color. But as always, you don't usually have my PowerPoints ahead of time anyways, right? Because it has all the answers. So, you know, take notes if you want to, right? If you printed it out, great, right? If you didn't, it's not the end of the world. You have access to it, okay? So, moving on to staining ID, and we're going to go over, you know, basic stuff about the exam as well. So for the exam, you need to be able to identify stains, and you've already kind of done this on your quizzes. And as I'll say later, your quizzes are excellent study guides, right, of the type of questions that you will see on our practical exam next Thursday. So simple stains versus negative stain. So this one is our simple stain. We know this because the cells are stained themselves, and typically we use basic dyes. The other reason is, what charge do bacterial cells have? What charge do cells have? Negative, right? They're negatively charged. What in their cells makes them mostly negatively charged? The DNA, right? The DNA. So they're highly negatively charged. So in this picture, right, the cells are stained with a basic dye, right? So some basic dyes we use are crystal violet, methylene blue, safranin. Just about all the dyes we have in our staining tray are basic dyes, right? There's only 2 exceptions. They're the most widely used. Because what charge do they have? They're positively charged. So they stain the cell, but why? The cell is negatively charged. The dye is positively charged. What do we know about these opposite charges? They attract, right? Remember the old saying, opposites attract, right? I always say that. Explains my first marriage, right? Opposites attract. Make sense? Right? So that's why, right? The positive is attracted by the negative. It goes into that cell. And it colors the cell. Unlike what you see here, right, an acidic dye was used to do negative staining, which this kind of helps you too, because the acidic dyes that we have in lab are Congo red and nigrazine, but another one that's sometimes used by microbiologists is eosin. These have what charge? They're negative. Negative staining uses a negatively charged acidic dye. Now, though, we've got negative cell, negative dye. So in this case, the background gets stained because what about those like charges? They repel each other, right? So like trying to stick 2 magnets together and they keep flying apart, you've got the 2 like poles coming towards each other. All you got to do is flip it, and then they'll stick. Because at one end, it's positively charged. At the other end, it's negative. If you've got the 2 positive or 2 negatives trying to come together, the 2 magnets literally just keep flying apart. All you got to do is flip one of them, so you've got plus and minus coming together, the opposites. So like charges repel each other. In this case, negative and negative. So you should be able to recognize, right, when you see the background stained and not the cells, that you're looking at a negative stain. If you see the cells stained, you know it's a simple stain with a basic dye. What color those cells are might tell you even more information, right? As we learned last week when you do the gram stain. Because we actually use 2 dyes, and we have a decolorization step. The first dye we use, crystal violet. Then you rinse with distilled water, right? And then we stain with grams iodine. Another thing I would have fixed Tuesday if I was here was better labels on our iodine bottles. But now that's gotten, washed away. It's going to have to wait. Then we're going to decolorize. What do we use as our decolorizer in the gram stain? Alcohol, we use ethanol, right? The particular alcohol we use is ethanol. But either is usually acceptable, right, for testing purposes. If you write alcohol, that's a general name for ethanol. Ethanol is the specific alcohol that we use. It does make a difference. You can't use isopropyl. You got to use ethanol. Okay? We do this dropwise over a paper towel until it goes from dark purple to light purple. You don't want to do it until it's clear. You'll over-decolorize your gram positives, right, and that won't give you accurate results. In order to see our gram negatives that have become decolorized by the decolorization step, we do our last counter stain with what dye? Safranin, which is a red dye, but the cells will appear pink, right, and that's due to the damage. So we rinse with water. We blot dry, we do not rub, and remember we're looking at bacteria, so always from now on, always oil immersion, all the way to 1,000 times magnified. So what color are these cells? They're purple. What dye made them this color? The crystal violet, and therefore they are gram positive. Remember, does this have anything to do with charge? No, this is a result in the gram stain reaction. It stayed purple, so therefore it's positive. What color are these cells? Pink. What dye made them that color? Safranin. And so what are they? Gram positive or gram negative? They're gram negative. And again, this is because they got decolorized and got stained pink. Safranin is a red dye, but because of the damage these cells go through the process, they don't retain a lot of the red dye, so they're a lighter shade of red, which is referred to as pink. And that is important, right? They are pink. Some people say red. You'd have to preface that with a lighter shade of red, right? It isn't true on red. We will see, hopefully, true on red today. Okay, so why do we do the gram stain? One of them comes out purple, one comes out pink, and what's the big deal? So what? Yeah, two membranes over here, right? And this layer, what's this layer? This is the peptidoglycan layer. The structure is referred to as the cell wall, right? For gram positives, how many membranes do they have? Just the one cellular membrane, right? Sometimes referred to as the cytoplasmic or plasma membrane. I always just refer to it as the cell membrane. It's pretty simple, right? But just one. And then outside of that membrane, it has a layer of peptidoglycan. In comparison to gram negatives, is this thin or thick? This is a very thick layer of peptidoglycan. And remember, this makes a difference. This is what traps in the crystal violet iodine. When this shrinks, when exposed to the ethanol, it traps in the crystal violet iodine complex and makes these cells stay purple during the decolorization step. These guys, gram negatives, typically, on the other hand, bacteria that is, have how many membranes? Two. They have the cell membrane, and what do we call this one? The outer membrane. That one is pretty easy, right? It's the outermost covering. It's the outer membrane.
Sorry, the one last figure that you showed is there was the sugar and protein. Is that
Lipid and sugar, yes. So for gram negatives, one of the reasons why we would want to know, is it gram negative? This outer leaflet of the outer membrane, this substance is not phospholipids. It's lipopolysaccharides, which are toxic for us. Right? So notice the little squares here and then things sticking out of the cell, not like what you see here, right? Or here or here. These are all phospholipids. This is lipopolysaccharide. And this is one of the reasons why we want to know, is it gram negative? Because it has this toxic substance, this LPS. And so when we gram stain them, because they have this outer membrane, it deteriorates, right, with exposure to ethanol. And even though they have a peptidoglycan layer, it's what? It's thin. It's not enough to trap in the crystal violet iodine. So it gets washed out. They become colorless. We've got to stain them with safranin, which they're not going to hold a lot of that dye, but enough that we're going to see them as a pinkish color.
And the LPS completely dissolves?
For the most part. It creates a lot of damage to that outer membrane. So the crystal violet iodine just comes out, washes out. And that's why you have purple coming off your slide, right? Because crystal violet is washing out of those cells. So we also did the capsule stain last week. So which picture here has a capsule? A. Why do we need to stain the cell in the background around a cell to see its capsule? Because the capsule, one, doesn't stain, so we can't, use one dye and stain that. But also, where is that capsule located? It's the outermost covering of a cell if it has it. So if we stain just the cell, we won't see it because it's clear outside the cell. We've got to stain the cell and the background, and the capsules, we said, doesn't get stained, so we see this clear halo around the cells. So that's the capsule that's not stained, that's around the outside of the cell. In this picture, you don't see that halo, right? That's because they don't have a capsule, right? So it's cell right up to background. So it's located outside, right? What's the function? Why do some bacteria have this capsule? Why is it beneficial to them?
Protection and
It helps them stick. So adherence, right, allows them to attach to us, right? And it protects them. Let's go one step further with the protection. How does it protect them?
Prevents phagocytosis.
Right, protects them from phagocytosis. Our white blood cells normally would eat them up. But because they have this extra covering, it's like they're disguised. Our cells can't see them to be able to attach to them and gobble them up. Right? Our body would have to make things like antibodies, proteins that will specifically bind to that layer to be able to recognize them. And that's actually what some of the vaccines that we get help us do. Whooping cough, right, caused by pertussis. That's an encapsulated bacteria. It has a capsule. They actually give you in the vaccine components of that capsule so that your body will make antibodies against it, and so that way you will bind, those antibodies will bind to it, your phagocytic cells can find it and gobble it up and eat it and destroy it. So having a capsule is very beneficial to bacteria, right? It helps them attach to surfaces, and we're talking about biotic surfaces, right? So living things. Not our slides though, right? That was one of the problems we ran into, is they don't stick real well to glass slides. That's why we didn't rinse our slides. Because we didn't use the serum to help stick them to the slide. We didn't use some protein. And that's basically there's a lot of protein in the liquid phase of your blood, right? That's what serum is, except serum doesn't have the clotting proteins. But it has all your antibodies and a bunch of other proteins like albumin and such. And so they get real sticky. And then, of course, protection, and, that's against being destroyed by our immune system. Today you're going to do the acid-fast stain. And for the acid-fast stain, again, it's a differential stain. So just like gram stain, we're going to have what step?
Acid. Yeah, alcohol.
The acid alcohol. What does that do? What does the acid alcohol do? It's a decolorization step. So it helps us differentiate acid-fast positive, or what we just say acid-fast, as opposed to acid-fast negative, or what's commonly referred to as non-acid-fast. So same thing as the gram stain, right? We're going to start out with a primary stain. In this case, it's going to make the cells a purplish pink color, what's often referred to as fuchsia. So a reddish purple color. And the dye that made them this color is carbofusion. There are two different widely used methods of this procedure. We're going to be using the Zeeland-Neeson method, which is commonly abbreviated the ZN method. In that method, the carbofusion that we use isn't as concentrated as the Quignon method. And which is good for us, y'all, because the really concentrated stuff is very smelly. It contains phenol, right, which is a substance that's really smelly to us and can be damaging for us if we were to inhale large amounts of it. So the good news is lower concentrations in the one that we use, so we don't have to be in a fume hood because we don't have one in this lab. But because we're not using a very concentrated dye, it makes it a little bit harder for that dye to stain those cells. To help facilitate that process, we're going to heat the slides while we're staining them with the carbofusion today. And so that's why we have our heating apparatuses set up today. So if they stay that color after decolorization, right, with that primary stain carbofusion, they're said to be acid fast, right, or acid fast positive. What color are these cells? Kind of bluish gray, right? Or sometimes a greenish blue or a grayish blue. Depends on which counter stain you use. And obviously you're going to want to use some stain that doesn't look like the carbofusion. So we're not going to use a red stain, right? We're not going to use a purple stain as a counter stain because we might confuse that with being acid fast. So a lot of the different stains that we use are a bluish or greenish stain. So some procedures call for brilliant green. We found that brilliant green isn't so brilliant under the microscope. And we've had better success with methylene blue. And I think the newer protocol that's in your book, right, says methylene blue for today, I hope. Your acid fast stain. And so if they don't say the carbofusion color, they get decolorized, and then therefore they would be said to be non-acid fast or acid fast negative. What you got? Yeah, methylene blue. I thought so. Yeah, we got rid of the brilliant green because, like I said, it's not so brilliant anymore. When I was doing research, leprosy research, we used the brilliant green. We didn't really have any non-acid fast. We were looking for acid fast. Just in case. So again, as it relates to spore stain, you need to know what method you're using and what dyes you're using so you know what color things are supposed to be. So we're going to employ the Schaefer-Fulton method for endospore staining. And in that method, what color are endospores supposed to be? They're supposed to be green. Notice I said supposed to be. What dye made them this color? What dye are we going to use? We're going to use malachite green. Malachite green is kind of an aqua green, like a bluish green. And I find it tends to be more bluish lately than green. I don't know why. And so our cells, even though they're supposed to be green, they look a bluish green. Does that make sense to you guys? Like an aqua bluish green. Yeah. They're still beautiful, though. They look like little gems, you know, little gems under the microscope. So, from your homework, there's some terminology that we use for describing the location of the spore when it's still inside the cell. The spore forms within the cell, so that's why it's called an endospore, although some scientists like myself a lot of times just abbreviate and say spore, which is bad because other organisms do produce spores, right? And it can be confusing. But with bacteria, we're talking about endospores. And these form within the cell, and they'll form in the same location always for that species. So in the case of this one, you can see it pretty well, right? You see the spore, you see the cell. What term will we use to describe this location of the endospore? This would be subterminal. So it's not completely at the end, right? And it's not in the middle. At the end, it would be what? Terminal. It right smack in the middle, central. And this is in between those two, right? So it's subterminal. The good news for you guys is underneath the microscope, you won't have to do this level of identification. We might get lucky today, and we might actually see endospores still within cells. But as you guys have already experienced, these bacteria cells are really small, right? Some of them. And then we're trying to locate something inside of them and determine its location. Next to impossible, right? We might get lucky. We might see, right? You can kind of guess by one of the bacterial names, Bacillus subtilis. It's subterminal. Its location tends to be like we see here in this picture. But again, it'd be extremely rare. It'd be cool. We might get to see it today. But obviously on the practical exam, I'm not going to set up under a microscope that slide and point to a single bacteria and ask you to tell me the location of the endospore. It's just not practical, right? But I can throw a picture at you like I did on your quiz today, right? And you need to identify it that way. So if you did all your homework, that was easy, right? Oop, I gave you the answer already. Do you see any spores in this picture? No. And what color are our cells going to be using this staining procedure? Yeah, these cells. The cells themselves. They're supposed to be red, right? I know I admit that this color is more orangey, right? I stole pictures from the internet, y'all. And the color just wasn't quite right. But no green structures, right? That's the important thing here. No green structures means no spores. So under what conditions are spores going to be formed by an organism that can do this? Unfavorable, harsh. Guess how I got them to do spores for you guys? I starved them. Yep. So I put them in those tubes Tuesday, right, before I had to jet out of here. So they've been in there for 48 hours now. That's actually a pretty long time. They're running out of nutrients, right? So a lot of them have started forming endospores. Other things will make organisms form endospores. Changes in oxygen levels, lack of water, radiation, UV radiation. Lack of food is a big one, right? And the easiest one for us to do to them in lab, to make them form spores so you can stain them and look at them. So let's see. In picture A, what do you see? You see the green endospores, don't you? And the red cells. So what stain is this? It's an endospore stain. What method? No. ZN is for acid fast. Schaefer-Fulton method for endospore. Picture B, what do you see? What color is the background? Background stained in this picture, yeah? Are the cells stained? What color are those cells? They're purple. So the cells are stained, the background stained. This is probably what? It's a capsule stain. But what don't we see in this picture? A capsule. So this is a capsule stain, but it's negative. But the reason why you know it's a capsule stain is both the cells and the background are stained, even though that this is capsule negative. Next picture, what do you see?
I think that's just like grand stain.
But are you truly seeing pink and purple?
Yeah.
What are you saying is purple? Because look down here, y'all. Do you see purple here?
Oh, yeah.
That's definitely purple. Do you see purple up here?
Bluish purple.
Do you think, what are the dots? The dots are more bluish, I would say, right?
Yes.
What about this stuff? this clump of rods. What color would you say that is? Pinkish or reddish, right? And there's a clue here. Acid-fast bacilli clump together. You see how the bacilli are clumping? And the fact that they're a pinkish purplish color, right? This is an acid-fast stain. And that's the clue for you. The clumping of the cells that are rod-shaped, when you see them individually, you see the rods? And that they're that pinkish purple color. And then if you're seeing some other bluish or greenish or grayish color, those are your non-acid-fast cells. This is an acid-fast stain. Now down here is definitely purple and pink, right? So this is definitely gram stain. Here, what is stained and what is not stained? The background stained and the cells are not, right? So this is what? This is a negative stain. Done with probably nigrazine, right? Because it's a grayish background. If it was Congo red, the background would be red. So, negative. And the clue here is the cells are not stained, just the background. Okay, we gotta know shapes. So let's review. What are these? Cocci. A single one is a coccus, right? What's the term for pairs? Diplococcus. Groups of 4 in a square of cocci. Tetrads. A chain of cocci. Strepto, right? Remember strep throat. A packet of 8 in a cube. Sarcinia. This C sounds like an S because of the I. Yeah, I'm doing that again right now with my son. It's fun. Phonics. Gotta love it. Cluster. Staphylococcus, right? Because these are all cocci. Okay. Rod shape, what's the term we actually use for the rod shape? Bacillus, or bacilli if we're talking about all of them. So again, we have pairs, so it's diplo, but they're rods, so it's diplobacillus, or bacilli if we're talking about all of them. And then a chain of rods, strepto. Remember, chain is strepto. These are rods that have a little curve to them. This one also has a flagella. But what's the shape of this cell? Vibrio. Multiple curves. Spirillum. What's the difference between a spirillum and a spirochete? Spirillum, right, are these wavy lines, right, and they're really curved. Big curves, right? If you were to look at them three-dimensionally, though, they're pretty much flat. How are the spirochetes, though? They're helical, like a spiral staircase, right? That's the difference. And then under the microscope, I have that picture next. They look like zigzag lines, right? They're much tighter than spirillum are. And then if you can't hold a particular shape, because you're probably missing a cell wall, you're said to be what? Pleomorphic, or the condition of pleomorphism.
How do you spell spirochete again?
It's right here. Right? So these guys are spirochetes. And you see how it look like zigzag lines? And this is due to their axil filaments that help them form this characteristic shape and allows them to literally corkscrew through their environment. Right? They spiral themselves through. Spirochete or spirochete. It's pronounced both ways. Like tomato, tomato. We're all in bacteria right now. Yeah. Eukarya are long and gone, but not as far as the test goes, right? All right, so all exercises we've covered so far, including today, is on the exam. So 1 through 6 labs. Your quizzes are a good example of the type of questions I will ask on the exam. So make sure you review those. The quiz you took today, you'll notice it was all multiple guess, so I can quickly grade it and give it back to you. So you'll have that to study as well. You should know all the terms you were assigned for homework, right? All terminology. This includes being able to, of course, spell them. It's not going to be multiple guess on the tests. There's going to be a line, right? Standard practical exam form, right? You will write the term on the line. You need to spell it enough that I recognize what it is, so you can get your points, right? If I don't know what it is, it's wrong, right? If it isn't sufficient, right, but I can figure out what it is, you'll lose a point, right? You'll get half off. Okay. So if you don't want to lose any credit, spell it right. And write neatly. If I can't read it, it's wrong, y'all. You got to bring your best penmanship that day. And I always suggest bringing a pencil with a good eraser, right? Because pen, you got to scratch out and then you run out of room, right? So bring your pencil with a good eraser. And I also have erasers and little stubs of pencils too, if need be, if you forget. There will be lots of slides you will have to identify. Both eukaryotic organisms, right, and bacterial stains. As we've been going over, right, the bacterial stains, right, and then you have those practice quizzes I highly suggest doing for reviewing eukaryotes, because they're on the test. Of course, not all, 15 of them, 14, I can't remember how many there are. Not all of them are going to be on the test. About half of them will be, though, right? 5 or 6 of those, you don't know which 5 or 6, are going to be on the test. And all the questions you've answered in your homeworks, right? So we kind of did a little practice quiz in class, too, with the microscopes, where, it was pointing to the fungus Aspergillus, and you needed to be able to name it. You need to know that specifically it was pointing to the asexual spores. And then one of the questions we asked about Aspergillus was what commercial compound is produced by this organism. You would need to name one of these, right? Citric acid or soy paste or soy sauce. Anybody remember which mold is found on your bread? Starts with an R. Rhizopus. Rhizopus. You need to know all the steps to all the staining procedures and why you see what you do. Right? This staining ID assignment will help you, as well as the additional quizzes, the practice, and the quiz 6 that I put on Canvas for you. You need to be able to identify what staining procedure, right? So what are you looking at? What procedure might this be? And obviously there'll be enough information there that you'll be able to figure it out. And then you need to be able to identify, results. If you're seeing spores, are you seeing a capsule, right? Be able to draw and label that. Yes, you will have to draw for this test a few things and label them. Mm-hmm. Yep. You should be able to distinguish, as I said, gram-positive from gram-negative, right? So if I put a slide up there and you see purple cells, you tell me gram-positive. You see pink? Gram-negative, right? Same thing for acid-fast, spore, all of that. You need to be able to look at a slide, and if I even tell you, hey, this is an acid-fast slide, are the organisms it's pointing to positive or negative? You need to be able to tell me based on their color, right? In the case of acid-fast, there's another clue. They're going to always be rod-shaped. That's why you see the common abbreviation AFB. Stands for acid-fast bacilli. Not bacteria. That's okay. All right. So, the organisms themselves that we've worked with, right? We've been recording a lot of information about them. Mycobacteria we're going to work with today. And the reason we're working with it today is because it's acid-fast positive, right? And as I said, their shape is always what? It's always rod-shaped. And their arrangement isn't a common arrangement we see, but they clump together. And that's really an artifact of staining, more so than how they exist in nature. E. coli, right? What shape is that one? Do you guys remember? You gram stained it last week. It's a bacillus. It's rod-shaped. Does it have a characteristic arrangement? Do the cells stay next to each other when they divide? No. They're what we call singular. When you stained it, it should have came out pink, so it would be gram negative. Acid-fast reaction. Only mycobacteria come out acid-fast as far as bacteria go. Most bacteria. Most rod-shaped bacteria. So having known that information, any other bacteria I have on this list, if we were to stain it, it would be what? It'd be acid-fast negative or non-acid-fast. Right, yeah. Scientists don't say acid-fast negative, they say non-acid-fast. The reason why they're doing the acid-fast stain is they want to see if they have acid-fast organisms. If they have AFB. If they do, then they just say it's acid-fast. If it's not, they say that. They say it's non-acid-fast. But you could say acid-fast positive and negative. That's acceptable too. Just letting you know that scientists don't use that terminology. They say acid-fast or non-acid-fast. Okay, so Micrococcus luteus. So how does this relate to the test? I could say, this is Micrococcus luteus. Tell me what its shape and arrangement is. Right? So you need to know this. So for Micrococcus luteus, you should know it is... It's the sphere, so it's cocci, right? And its arrangement is very unique, right? Tetrads. And this one we gram stained. It came out purple, so it's gram positive. And if we were to acid-fast stain it, it would be negative, non-acid-fast. Staphylococcus epididymis. What shape are the individual cells? Its name tells you, right? They're cocci. And what's its arrangement? Staph is clusters, right? Gram reaction, they would come out purple. So they're positive. In acid-fast, they would come out negative. And that's the one you're going to use today for your acid-fast stain. Klebsiella pneumoniae. What special structure does this one have? Why did we stain this one last week? Cause a really bad pneumonia. It has a really big capsule. What shape was it? It was a rod. If we were to gram stain it, it would come out pink. So that's gram negative. It doesn't have a characteristic arrangement. We would say it's singular. Doesn't stay stuck. Today, and we've worked with one of these bacilli previously, but again, the name of the genus gives you a clue. What shape are these? These are bacillus. And they tend to form chains, right? So their arrangement is chains. So hopefully we'll see some chains today. And why we're using them today is they too form a special structure that we want to see today. And that structure for today is what? Endospores, right? We're using all these bacilli because they're all endospore formers. That's their special structure. If we were to stain them with the acid-fast stain, they would be everybody's going to be negative except who? The Mycobacterium. Oh, I forgot I had all of them like that. It's alright. You guys will have it because I'll post it. This is my last slide, right? Yep. Okay. So, I want to keep these as two separate recordings.